of the fundamental answers to the questions about its biology, right? But unfortunately, we didn't know what was happening right here. What is this organism doing in this image that you see here? The question of how it feeds, what it's feeding on, was a mystery for quite some time. And so as a graduate student, I looked at this problem and thought, well, there are three things that I would expect to see if this parasite is feeding on the bee's blood. Because all of the studies, all the publications, all the posters, all the fact sheets say, varroa destructor feeds on the blood of bees, nothing else. That is what they consume. But I would have expected that their digestive system would be structured like the digestive systems that you see in other creatures that feed on a food product that is exclusively fluid. I expected that they would be closely related to other organisms that feed exclusively on fluids, and I expected that they would feed in a variety of locations on the bee's body. The way that a mosquito or a tick can draw blood from pretty much any surface on you because blood is ubiquitously distributed, you would expect that varroa wouldn't just have to feed in one spot on a bee's body, but if they are feeding in one spot, they may be feeding on a tissue exclusive to that location. And that tissue turned out to be the fat body. As I went through these expectations and it was clear that none of them were met, I then biostained the fat body and the blood of the bees with fluorescent biostains. One of the coolest things that you can do as a scientist is solve a problem with glowing fluids. It's everybody's goal. If you're a child who wants to be a scientist, that's all you're thinking about. So I got to feed these bees glowing biostains in their sugar water and stain the fat body. So this is the abdomen of the bee, and all of that yellow glow that you see there is from the hemolymph. And then the fat body of the bees was glowing red. And so the expectation was if the mites feed on these bees and they consume the blood, they'll glow yellow. And if they're glowing red, then they're feeding on the fat body. So this was an exciting set of images for me to acquire in that process. And then I conducted studies of survivorship to determine if they needed it for the production of offspring and for them to be healthy, and it turns out they did, very much so. This was actually the decoy bee that I constructed to feed them these different food products. It looks pretty low budget, but that took a lot of work back in the day, you know? Size 5 gel capsule, same size as the pupae and the larvae. I thought it was really cool. But anyway, we were able to determine through those studies and through looking at the spatial location of the mites on the bee's body and how they only fed in the area where the fat body was present, that they were feeding on the bee's fat. And that fat body tissue is incredibly important to the growth and development of the honeybees. There are 9 essential functions that happen in the fat body, many of which happen nowhere else in the bee's body, and they are necessary for the bee to grow and develop properly. And when that tissue is damaged, bees end up with a lot of issues. So you can probably guess that after acquiring images like this at the United States Department of Agriculture, where we were able to section all the way through the bee's body, the mite's body, see the internal contents of both organisms, and really get a feel for what was happening with this creature, you can probably guess that after studies of this nature, the wheels really started turning as to what this really meant for our bees. Because we've been thinking since the late 1960s, early 1970s, that varroa destructor feeds exclusively on a renewable resource in the bee's body. They climb onto a bee, they feed on the bee's blood, they go away. Well, blood is constantly renewed in the bee's body, so the expectation there is different from the expectation that you would have if the creature is feeding on the bee's fat body. Now, the fat body is analogous to the liver in a mammal. So let's think about it for you. If a mosquito landed on you, sucked out some of your blood, that'd be concerning to you, right? You wouldn't be your favorite part of the day, but you'd get over it. You make some more blood, all be right with the world. Now, if the mosquito landed on you, liquefied your liver, and then sucked that out of your body, different implications for your day, I would guess, right? Are we there? We're on the same page, correct? Okay. So this work was published in the Proceedings of the National Academy of Sciences, which was a huge honor. I was very excited about that. It even got to be the cover article. Not bragging, but I was super excited. And then the wheels really started turning. So we've discussed the background here. We've discussed how I became the fat body guy. But there was something really interesting going on in this image up here. You can see varroa destructor feeding on an immature drone. You see this white viscous substance, liquefied fat body, going through the mite's body. But the question that kept coming up for me is, what are they doing with that ridiculous amount of nutrition in the fat body tissue? They have targeted a tissue that is incredibly nutritively dense. It is the storage site for all of the stored nutrients in the bee's body, and the storage site for a very important set of nutrients, the bee's egg yolk proteins. Now, egg yolk proteins are called vitellogenins, and every creature that produces eggs has to create something to sustain that embryo as it's developing into an adult. And it doesn't just need energy, it needs the full complement of amino acids necessary to structure this new organism, right? So vitellogenins are really important things, and what I have been thinking, as I've looked at Varroa destructor, and guys, I've taken so many pictures of this creature via light microscopy, electron microscopy, confocal microscopy, using microscopes I'd never even heard of before I started my graduate program. I've seen these things from every angle. This is one of the most striking images that I've ever acquired. That egg, by comparison to that mom, is huge. It is about 41% her body volume. And she is able to make an egg that size every 30 hours. That is staggering. That is a feat in science. Now, usually when I would talk about this, I would say that's like an adult woman who's 100 pounds having a baby and every time someone stops me, it's like, nope, I don't want to hear it. No, do not finish that sentence. That is deeply disturbing. But this mite is able to go through that process every single day. Now, for most organisms, the amount of work that's required to produce that much vitellogenin, that many egg yolk proteins, is too taxing for them to consistently go through that process and create that much vitellogenin that quickly. So I wanted to analyze these eggs and see exactly what's going on inside of them. And so we took eggs from several mites, and it took a while to get all of these eggs. We were trying to do this in Maryland, and I couldn't get all of them in time to finish this project. And I went to ABF and was talking to a group of people there, and they're like, oh, we're non-treatment beekeepers, and but we don't have any mites. And I was like, you don't? Why don't I come down to Florida and hang out with y'all for a little bit? We'll see. I went through one colony and was able to find all the eggs I needed to run all of these assays. It was amazing. But part of what you can really see here is that, especially bees that have some level of scutelinization, so the ones that are from Apis mellifera scutellata, oftentimes have pretty substantial tolerance to varroa, and so the mites their populations can blossom to ridiculous levels, and the colony can still survive the experience at times. It's quite remarkable. So we're able to collect these mite eggs and then run gel electrophoresis. And that's basically like putting all of these proteins through a maze. The proteins are electrically charged, and so you attach an electrode to one end of this maze that allows for these proteins to migrate all the way down the lanes. And then because the different doors of this maze are different sizes, the larger proteins sequester in one area and the smaller ones in another area, and you can separate them by size and determine what you have there. And the interesting thing that we found that we had here was that the same proteins, the same vitellogenin proteins found in the honeybees, let me see, is this working? Yeah? Oh. All right, maybe not. The same vitellogenin proteins that were actually found in the honeybees were found in the mites' eggs. Now remember that I told you earlier that every organism that makes vitellogenins, or every organism that makes eggs, makes vitellogenins. Well, every single group of vitellogenins is distinct in its sequencing. All of these organisms make a very different set of them because different creatures need different sets of proteins, amino acids, and fats in order to grow into adults. And so the fascinating thing here is that you never expect an organism to have the exact same vitellogenins. How did they get the bees' vitellogenins into their own eggs? Well, it got